Measure of biodiversity. Biodiversity means variation of flora and fauna. Variation of flora and fauna is measured in three levels. Ecosystem level. At this level of study, diversity of different ecosystems is studied. For example, aquatic ecosystem, desert ecosystem, grassless ecosystem, forest ecosystems. These are the different ecosystems may be present in study area. Next level of study involves the study of diversity in species present in each of these ecosystem. So for example, in grassland ecosystems, the species 1, species 2, species 3, and species 4 are identified. And third level of study is the genetic level. At the genetic level, the diversity of gene or the variations in gene is studied. So these three levels of variation can be understood by an example. So let us understand it. Suppose there is a study area, and this study area has got different ecological regions like forest, desert, grassland, and lake. Each ecological region represents an ecosystem. So these are documented as forest ecosystem, desert ecosystem, grassland ecosystem, and aquatic ecosystem. Existence of different ecosystem in study area is called ecosystem diversity. In the next level of study, species present in different ecosystems are documented. Existence of different species in different ecosystem is called species diversity. The third level of study is documentation of variation in genes. The variation in the same gene is called genetic diversity. To understand this genetic diversity, let us take an example. For example, there is a population of plant species growing in this forest area. Some individual of these species bear white flower and some individuals bear red flower. The presence of two different flower color is due to presence of two different alleles for the same gene. This means the gene responsible for the color of the flower is having two alleles. One allele is for the white flower and the other is for the red flower, creating two varieties of the same species. So variation in gene is called genetic diversity. So these three levels of variation are documented to describe the biodiversity of a given region. So once we document the biodiversity at three different levels, that is ecosystem diversity, species diversity, and genetic diversity, we need to calculate the diversity index. Diversity index is the quantitative measure of biodiversity, or here we can use the term species diversity in place of biodiversity. And the index is a numerical figure that explains the biodiversity of a study area. The diversity index is calculated using different methods. There are more than one way to calculate biodiversity index. However, we have mentioned only two very important diversity index. They are the Shannon-Weiner index and the Simpson diversity index. Both the indices are calculated by their respective formulas. The calculation of Shannon-Weiner index is based on two factors. One is species richness, and other is species evenness. Species richness is the number of species present in a given community, and the species evenness is relative number of individuals of each species in a given area. The species richness and the species evenness are calculated from the list of species and then easily put into the formula to obtain the biodiversity index. Shannon-Weiner index is influenced by the species richness, that is abundance of a species, and evenness of the distribution, that is the presence of dominant and rare species. So it is sensitive to even a small change in both the factors, and it is popular and widely used to determine the state of the ecosystem. On the other hand, the Simpson Biodiversity Index is based on the probability that two individuals collected randomly belong to two different species. The Simpson index is influenced by dominant species, so it is used to determine the dominance of species that shows the trend of the ecosystem diversity. Both the indices are used to obtain the information that are useful to measure biodiversity of a region. So there is one more way to measure the biodiversity. The ecologist often observed that the biodiversity is not evenly distributed. The biodiversity often exists in patches or confined to certain localities. So alpha, beta, and gamma diversity were introduced by R.H. Whitaker to measure biodiversity in a given area where the whole area is divided into some localities or sites or ecosystems. The total species diversity in a given area is called the gamma diversity. The gamma diversity is determined by two other findings. These two findings are the diversity in different locality or site or ecosystems, and that is called the alpha diversity. And the second one is the differentiating among these sites, that is the comparison between the two sites. It is called the beta diversity.